Good morning. I'm Jack Van Horn from the University of Southern California and the past program chair of OHBM. It is my honor and my distinct privilege to introduce our keynote speaker this morning, Dr. Susan Bookheimer. Dr. Bookheimer holds the Joaquin Fuster Chair in Cognitive Neuroscience and is a professor in the Department of Psychiatry and Biobehavioral Sciences at the David Geffen School of Medicine and the Department of Psychology at the University of California, Los Angeles. Dr. Bookheimer received her bachelor's degree in psychology from Cornell University in 1982 and her PhD in clinical psychology from Wayne State University in 1989. Following this, she undertook postdoctoral studies at the the National Institutes of Health before coming to UCLA in 1993. Among her many responsibilities at UCLA, she serves as the director of the Staglin Center for Cognitive Neuroscience and as well as the Center for Translational Research and Neurodevelopment. She has been an active member of OHBM since its inception, having served in many positions on the OHBM Council and served as the chair of our organization from 2012 to 2013. She is a clinical neuropsychologist whose work has spanned both basic research as well as clinical practice, with nearing 300 published scientific articles using brain imaging technologies. Clinically, she is responsible for conducting the functional magnetic resonance imaging and intraoperative mapping of critical brain functions using electrical stimulation of the brain in pre-neurosurgical populations at UCLA. Her neuroscience research program employs functional imaging specifically to try to understand how brain functions, alterations in brain functions give rise to the major symptoms of autism spectrum disorder, especially in the context of language, social communication, joint attention, and emotion. Moreover, her recent work has begun focusing on understanding the highly gifted brain and examines children with exceptional intelligence, exploring how the structure and connectivity of their brain develops and understanding the distinctive ways that the gifted and creative mind approaches and solves complex problems. She continues to contribute immensely to the understanding of memory capabilities of the healthy aging brain, as well as the changes in brain structure and function due to Alzheimer's disease and other degenerative processes. Today, Dr. Bookheimer will be presenting her 2016 OHBM keynote lecture on the uniquely human hippocampus. Please join me in welcoming Dr. Susan Bookheimer.
Thanks so much. Thanks, Jack, for that extremely kind introduction, and thanks to the program committee for giving me this opportunity to speak to you today. I'm going to be talking about the human hippocampus and thus memory disorders, primarily from studies that have been done from students in my lab over the years and also the work that they've done in their own lab since leaving mine. And I'm going to start with a case example like everybody does when they talk about memory, and I promise it will not be HM. Sue Corkin has already talked plenty about HM. Everybody talks about HM. Let us not speak of him again. I'm going to speak instead about one of the first patients I ever saw out of internship, which is a young man who had a rash and came into the emergency room with a fever and delirium. This was in the mid-'80s, and the young resident, in his wisdom or folly, depending on your perspective, immediately shot him up with acyclovir, recognizing he had herpes encephalitis and saved his life. When I went to see him, he looked at me in terror and said, "Who are you? Where am I? What am I doing here?" Explained he was in the hospital, that he had been sick, he was getting better, and I was going to do some psych testing with him. Psych testing? Yes, I'll be testing your memory. Who are you? Where I am? What am I doing here? And on and on and on, forever. It's very much like a famous case known in the literature from Clive Waring, who actually had the exact same problem, and this is a picture of Clive Waring right after his case of herpes encephalitis, which has a particular affinity for the hippocampus and basically wiped it out bilaterally, who's with his wife. And he has a similar reaction to his wife, but it's not of terror, of tremendous joy when he sees her, including when she leaves the room and returns, because he has no memory of her having been there seconds ago. But there's some interesting things about it. These are pictures of them over the 30 years that he's had this disease. And he doesn't say to her, "Wow, what happened to your hair color?" or "Why did you cut your hair?" or "How did you get so old?" or "My, haven't you gotten fat?" He continues to recognize her, so he's clearly learned information. But what he's lacked is this where he is in time. He's lost in a single moment in time. And he describes it as suddenly becoming conscious at every moment. And this is a note from his diary that I find particularly chilling. He writes, every few minutes, I do live. I wake for the first time. I am superlatively, perfectly conscious. I am perfectly awake. And he keeps crossing out his prior notes because he realizes that, no, that moment, that was completely wrong. It's this moment that he's conscious. And you can see that this is just one morning's notes. It's terrifying, but it tells us something that seems very unique about the human experience of having memory loss. When we talk about hippocampal memory deficits in humans, we talk about this loss of the continuity of consciousness, that you're lost in time. And the deficits are often described in extremely human attributes, deficits in self-awareness, conscious recollection. We speak about reference to specific episodes. We talk about declarative memories, which imply a language component. And these aspects of amnesia seem to be tied to very uniquely human abilities, which is ironic since the hippocampus is a very old structure and similar to that of rodents. In rodents, we talk about things like novelty detection, cell-specific functions in navigation, particularly place cells, grid cells, time cells, and some associative learning like food location, sphere acquisition, but certainly nothing like these processes that we see in human beings. So today I'm going to speak briefly about two questions that are related to this. The first is, how can we reconcile this uniquely human characterization of episodic memory with what we know of rat hippocampal function? So I'll be discussing three things: hippocampal circuitry, high-resolution fMRI of the hippocampus, and some single-unit recordings in human hippocampus. And then at the end, I'm going to spend a few minutes asking this related question: if the hippocampus is so utterly critical to what makes us uniquely human, and these amnesic patients simply cannot function without it, then why is the hippocampus so incredibly vulnerable to many disorders like encephalitis, anoxia, stress, seizures, and aging? And I'm going to suggest these are not unrelated questions. So I'll start by looking a little bit at hippocampal circuitry. These are pictures from the literature from Strange et al. Looking at the hippocampus of the rat, the monkey, and the human. And I just want to point out here, I guess it's hard to do that on everybody's board here, but they're very much the same, except in the rat it's kind of flipped around because his head's in a weird position. The entorhinal cortex is in blue, and if you flip it around, the ventral part of the hippocampus in the rat looks like the anterior hippocampus in the human and the monkey. And they have this neat internal architecture that's very sophisticated, with a central core of the dentate gyrus, and then these Kamens, the corpus amen, or Ammon's horn region, CA1, 2, and 3, and some people say 4, wrapped around it. And a circuitry has been pretty well worked out in a variety of animal studies using tracing studies. This is a hippocampal circuit from animals and a picture of the unique cellular organization in a rat. As you can see, it's quite a lovely and intricate organization. And you can see three general portions of this pathway. The first on the far left is from perihippocampal gyrus and pericrinal cortex, but everything from the outside, everything from the rest of the cortex enters through the entorhinal cortex. So there's a pit stop there before it goes into one of two loops in the hippocampus, one through dentate and CA3, which has these recurrent or reverberating circuits, and out, and another loop that goes directly to CA1 and subiculum and back to entorhinal cortex. So we want to look at this kind of circuitry using some human imaging. And one of my former graduate students, now a professor at Stanford, Michael Zaney, has done this recent, really beautiful work in postmortem tractography, showing those same structures very nicely and showing their connections. And you can see all of those same structures in the brain, the entorhinal cortex, subiculum, and so forth. Now, using the hippocampus to examine, using fMRI to examine hippocampal function has been very challenging. And there are many challenges over the years that we've dealt with, task challenges and others. I'm going to point at two of them. One is that there is functional heterogeneity within the hippocampal subregions, and the anatomy that I just showed you, I think, is good evidence of that. And there's really insufficient spatial resolution in most MRI techniques to resolve that functional anatomy. And what makes it especially difficult is that this hippocampus has an oddly rolled architecture, like what they say jelly roll. What's a jelly roll? Nobody eats jelly rolls anymore. It's like a Twinkie with jam in it rolled up, and they're really disgusting. So I would prefer to think of the hippocampus as more like a cinnabon, with delicious cinnamon goodness inside, along with brown sugar and nuts and all sorts of great things. And rolled up, it looks very much like the inner part of a cross-section through a high-resolution hippocampus like the one you see here on the right. And so to think about how to image that, if you wanted to look at functional activity in these hippocampal subregions and ask the question about whether they're functionally heterogeneous, you'd have to find a way to first get smaller voxels and get better resolution. And we had this crazy idea that we could really look at the organization of hippocampal function if we could essentially unroll that hippocampus, take the cinnabon, and lay it all out in one flat space and look at the organization all at once. It would look something like this. So the hippocampus in the cross-section has all of the neat and unique features that you can see, just as the cinnabon has like the dough layer and the brown sugar layer and the nut layer. But if you look along the long axis of the cinnabon, it's pretty much the same. So if you were to cut it the other way, you would get, for example, a nice long layer of dough, which would not be very appealing. But in the imaging world, it would be very appealing because since the structure maintains itself through the long axis, then we could look at voxels in an anisotropic way and try to get very small voxels within the plane and make up for the loss in SNR, because you always have compromise, by using very long voxels in the through plane, in the longitudinal plane, to regain some of that signal. So my graduate student, Michael, working in my lab, developed this program of segmentation and unfolding of the hippocampus that I'm going to show you now. That's him looking younger than he did on his most recent slide. And for the unfolding methods, we used what was the best technology we had at the time on a 3 Tesla scanner. And we used a T2 a fast Beneco pulse sequence because it has really nice contrast and also very little susceptibility in the areas that we worry about it. And we're able to get at that point a small field of view study with 0.39 millimeters squared in plane, and then make it up for that loss in signal by having 3 millimeters thickness that's perpendicular to the long axis. And so where we have the less resolution is all in tissue that has the same internal structure. And so this is what it looks like. The process involves first identifying the different subregions of the hippocampus, and we use that in reference to a standard atlas, although we're hoping to come up with better methods, more automated methods. We can't really resolve the CA2, 3 indentates. We're putting them all together in one region. But then we can delineate the outlines of the other regions, and then use the segmentation feature to first segment out white matter, then CSF, and then grow the gray matter in that ribbon in between. And this that you see on the lower left is basically the gray matter strip with everything else taken away. And so if you can imagine just taking that big strip there and pulling it out and folding it, you'd end up with a flat map that looks like what you see below. So you have anterior, where the anterior CA dentate gyrus area is, posterior, and then this is medial and lateral, and we bring it all the way out to the fusiform gyrus so that we can also look at some of the sensory input. So I'll show you some high resolution after my studies of the human hippocampus that we did early on. And the first one was a novelty encoding task. We chose this because the rodent data show hippocampal responses to novelty, and there's evidence of subregional heterogeneity in several of these regions in the hippocampus, including the dentate and the CA3, where those reverberating circuits are located. So we used a very simple block design, looking at new scenes and old scenes. It would be the same 2 scenes repeated, and the subject just had to look at the scenes and say whether they're indoor or outdoor. We did a bunch of blocks of those and then just contrasted the new from the old. This is the EPI sequence, so of course we can't get that great structure resolution in functional imaging, but we're at that point able to go down to 1.5 millimeters in plane. We can do better than that now. We normalized each block against its neighboring block to account for drift, and did a very simple contrast. And then because we have these flat maps, we're able to extract time series from each of the individual subregions and look and see how those individual subregions act. And so here is the results from that study. We have the right hippocampus over here and the left hippocampus on the right. And what I want to show you first is that the activity in the hippocampus is not spread all over the hippocampus. It's actually quite localized to a couple of small regions. I think I put little circles there. Yes, I did. And particularly in the very anterior CA3 dentate gyrus region bilaterally, and nothing in the CA1. Just a little bit in the subiculum, and then a whole lot of activity in the parahippocampal gyrus and in the medial fusiform gyrus, probably associated with the parahippocampal place area and the medial fusiform object processing areas. And we can further differentiate between these regions by looking at the pattern of activity over time. And we categorize these in three ways. Well, the CA1 and the entorhinal cortex showed no activation associated with a task. That's on the far right. And on the left, the regions that are outside the hippocampus but are responsible for the sensory processing aspect show these nice increases when they see new things and less activity when they see old things, which is, I think, you would easily expect. But what looks like learning is found in the hippocampus. It's an activation that develops over time, and that happens again in the CA3 dentate gyrus region and in the subiculum. So it supports the idea that there is heterogeneous activity patterns among these hippocampal subregions. We can't really treat the hippocampus as a structure as a whole if we want to understand human memory function at a more specific level. Associative learning is something that's also very important in the rat as well as in the human. In the human, we care mostly about associations that are highly relevant to us socially, like face-name associations. And so in this task that Michael published a number of years ago, we did a face-name association task where subjects saw faces paired with names, and then on a recall task saw the face, got a question mark, had to silently generate the name, and then we got their performance afterwards, and a simple distractor task. And we had multiple blocks of learning and recall so that we could look at learning over time. And this is a little bit busy, but what you can see is on the top we have right and left hippocampal flat maps during learning, and on the bottom during recall. And what I want to point out here is, first of all, in that same anterior CA3 dentate gyrus region that we saw in the last task, we see the same thing nicely during the learn condition. But if you look below in the recall condition, there's no activity there at all. And you'll also see that the activity in the fusiform and the parahippocampal gyrus has changed. It's now in the lateral fusiform area, right greater than left, again consistent with the face processing and possibly the word processing aspects of the study. So the sensory information localization has changed, but what's happening within the hippocampus, even though these are very different stimuli, has remained the same. And further, we see this nice dissociation between encoding and retrieval, where encoding we see primarily in this anterior CA2-3 dentate gyrus region, and the encoding changes over time, and it scales with the amount of new information that's being learned on any one block, so that by the end of the 4 blocks, the subjects have learned most or all of the associations. So the activity decreases over time, but notice that in that region, there is no activity whatsoever in the recall blocks, which are interspersed with the learning blocks. On the other hand, in the posterior subiculum, we see activity that's greater during recall compared to learning, but there is some activity during learning as well, which could involve some sort of re-encoding processes. And then in the fusiform, outside the hippocampus, we see what I think I would describe as a priming effect, more activity during that first block when they first see the faces, and then a sustained activity over time as they see the familiar faces again and again. So the pattern of activity is very different, and it suggests an encoding retrieval dissociation between these two regions, as well as a very separate pattern of activity that changes over time within the hippocampus as compared with outside the hippocampus. Now, I said before that we had trouble resolving CA3 and dentate gyrus at 3T, but we had the fortune to work with Bob Turner's group in Leipzig on their 7T magnet, and those were studies done by my former graduate student, Nantya Sutana, who's now an assistant professor at UCLA. And on 7T, we were able to reduce the fMRI voxel size to 1 by 1 by 2 millimeters in plane, and 1 by 1 in plane, 2 millimeter thickness. And this allowed us to differentiate the dentate from the CA2 and 3, and we used a very similar face name encoding retrieval paradigm, except that the names are now like Hans and Dieter and Günther and things like that. So we did it in Germany instead of John and Mary. Here are the nice structural images at 7T, and you can see that you get quite a bit of detail here. And on the right, you can see the breakdown of the subregions, and in red in particular, you can see that we're able to differentiate dentate from CA3 in orange. And that was really the goal of this study, and also to replicate the posterior subiculum findings. And so here's a more fancy flat map because it's got more regions in it. But if you look at the time curves below, you can see pretty much the same finding, except that we now know that the encoding activity, the learning activity, is associated primarily with activity in CA3 and not in the dentate. Whereas recall, once again, is seen primarily in the subiculum, although there is some learning activity in the subiculum as well. And again, we think that's because of the relearning, re-encoding that occurs during the recall process. Okay, but what about something that's like really uniquely human? Conscious recollection of episodes. And this is something that's been tested extensively in a cognitive paradigm called the remember-know paradigm. And in the remember-know paradigm, you learn a bunch of items, and then you're asked to retrieve them, to recognize them as one you've seen before and one you haven't. And when you've made that decision of items that you remember, there's a second step where we characterize items as items that were clearly episodically remembered. That is, that they can go back to the learning episode and remember it and provide explicit details. Like, I remember very well that at OHBM in Hawaii, I arrived one day dressed identically to my graduate student. I remember exactly what we were wearing. As opposed to no, where it's a correct declarative memory, but we only have the feeling of familiarity. Oh, I know that face, but I can't remember where it comes from, can't remember where I met them, but I do know it. I know that I've seen that person. And people can be trained to make these dissociations. And so one of our former graduate students who's from the Knowlton lab, we collaborated with, Laura Eldridge, did this study. She actually did a series of studies using fMRI during encoding and retrieval of object-object or object-word pairs. This is an event-related design, and I'm going to just show you the design real quick. The top is encoding, and the bottom is retrieval. So in coding, they would see these pairs of objects with words below, and the objects are oddly colored or placed in different positions, either on top of, for example, or next to the bread. And then during retrieval, they would see a cue word, and they'd first be asked, was this a word that was on the list? And they'd say yes or no. And then, if the answer was yes, then they would be asked, do you really remember the encoding episode? Do you really remember it, or do you just have a feeling about it? And we would score them, and then, therefore, average the time series for items in each of these categories. So here, I'm going to show retrieval at the top. Actually, this whole block is a retrieval because you can get both encoding and retrieval information out of the same block, because we have 4 kinds of stimuli. We have stimuli that are episodically remembered. So those are the R stimuli in red. And we have stimuli that were correctly remembered, but only with a feeling of knowing, without an episodic memory. And then there are lures, and then there are forgotten items. And what you can see is, if you look on the top row, in the left and right subiculum, and also to a smaller extent in the right CA1, that the hippocampus, in that part of the hippocampus, responds only to those items that it episodically remembers. So items that it actually truly remembers, and they're true memories, are processed as if they've never been seen before in that part of the hippocampus. In contrast, items that are brand new, for example, the correct rejections, the lures, show higher activity in the CA2-3 dentate gyrus region, that same area that we've shown before for encoding. And in fact, in the right hippocampus, there's also activity in the items that were completely forgotten. So it's as if the hippocampus, again, is processing them for the first time. Now, we could think of also doing the same kind of thing in a task that's a little bit closer to what we would do with rodents, like this navigation task. And this is a study that was done. We did a series of studies on navigation, and this is one that Nanthia has done, where you're navigated through an artificial city to some stores, and the encoding is you view your navigation, and the retrieval is you have to say whether the store is in the right place or whether they put it in the wrong place. This is done in a block design with the same encoding retrieval blocks that we talked about before. And here we've done a direct contrast between encoding and retrieval, so we've gotten rid of all the sensory information. But you can see, again, two things. First of all, that exact same dissociation between the anterior CA2-3DG region and the posterior subiculum for learning and recall, respectively. You also notice here that the stimuli are lateralized, and lateralization is something that we do find in humans, and lateralization of spatial navigation is thought to be a right hemisphere process in humans. So if we looked over a series of these studies, we see very consistent finding, and that is that the activity in the hippocampus is very principled. It seems to be in the same place during encoding, regardless of what the stimulus input is. It doesn't care about the content. It cares about the process that's being conducted, and that's the encoding process. And then we tend to see during retrieval activity in the subiculum, and again, it's content independent, but it's the same process no matter what stimuli we show. So most of rat studies, as you know, are done as single unit recording studies. So you can do these in human beings. Most of the rat studies involve place cells and various variations on this. This is an old movie of a rat navigating through an environment, and on the left are a list of different cells. And when he gets to different places in the environment, you can see he lands in areas where specific cells respond to that specific location. So he's got a group of cells that respond up top at yellow and a group of responses right there that look pink, and so forth. So that's what place cells are. You can make a heat map of that, and it'll look something like that if you track the rat's trajectory. So he's been running all around the place, and this particular one place cell fires there. There are lots of other kinds of cells that are variations of this that are much more complicated, and I won't have a chance to talk to you about that. But you might ask the question, well, geez, these cells this is very different from what we study in humans. But in fact, it has been studied in humans, and one of the first studies was done by my former postdoc, Arne Ekstrom, and this was actually done during his graduate work in my Kahana's lab, working in collaboration with Itzhak Fried at UCLA. So Dr. Fried and one of his colleagues developed this electrode that's used for recording seizures in patients with epilepsy. These electrodes are implanted, and they added a little special feature of these little tendrils. I don't know if you can see them here, but they stick out of the electrode, and they can go directly into neurons. And so they have the capability of recording individual neurons in the human hippocampus. And they record these in patients where they don't know the localization is, so they put them in both hippocampi, and then they use data from the healthy one. And so Arne did a similar spatial navigation task where he had an imaginary city and people navigating two different locations within the cities. And sure enough, on the right, you can see a heat map that looks exactly like the place cells that you see in rodents. So there doesn't seem to really be any difference. Something in single unit recording that may be a little bit closer to human beings is here's a study that was recently done by Nanthia Sutana now in her own lab, but done in collaboration with Itzhak Fried and I, looking at faces and lures. So here she's found a neuron that responds to Bill Clinton. And so we've got a picture of Bill Clinton. He's the target face. And then you encode these other photographs, and one of them is the one he's initially coded. And then there are lures of various similarities. So some lures are very close, like lure number 1, and some lures are very distant, like that's still Bill Clinton, but don't look a lot like him. And then there's another foil that would be someone, he who shall not be named. And there are... well, it's not as bad as Donald Trump, I guess. I could have said Donald Trump. We haven't found any Trump cells anywhere. And here are some other neurons that have been found. There's the Anne Hathaway neuron above, the Bill Clinton, and then there's a Jay Leno neuron. And if you look across, you can see that the greatest activation is to the original item, the original picture that's shown. And there is still some firing, but the firing lessens as the similarity to the target becomes less and less. Now, this is a process that's also known as pattern separation. It's been studied extensively by some other groups, particularly in Craig Stark's lab. I'm not going to really talk about that just for the interest of time. But what I'd like you to think about is... Well, first, she concluded that high memory performance is associated with more hippocampal activity, and in the same two regions that we've been talking about, CA2-3 dentate region and in the subiculum. But I think the question that you might all be asking is, do we really have Leno-specific cells, right? And not only that, do we really have specific cells in our brain that's for Leno in that particular tuxedo? And of course, we don't. We couldn't possibly. And the probability of even finding a cell, given the very small number of cells we're able to record that responds to any specific person, argues against any of these cells being specific for the content of stimuli that's coming in. Rather, the activity has to do with a learning episode in real time. That is, it's the sensory input of Leno at the time of the testing episode. So it's really the episode that we're measuring, not the Leno that we're measuring. It just happens to be that information from Leno is coming in, and we've got a cell that happens to be firing at that time to indicate this is the time, this is the episode in which we are studying J. Leno. And it's consistent also with this concept of remapping that's found in rodents, in human place, as well as in human place cells. That is, a place cell for one navigation environment can form a very different role in another navigation environment. They remap. And I would suggest that this suggests, and I think Arna has written a paper recently that argues coherently that, cogently, that these place cells also reflect the episode, that they're not really place cells. It's a language that we use to describe them, but it's not really that they're place specific. They're responding to the input at that moment in time. So if we look overall at our circuit diagram, we can draw some conclusions just within the encoding retrieval domain. The fMRI data appear to replicate the circuit functions that are hypothesized in the rodents. So pre-entorhinal cortex, so the rest of the cortex sending information into to the hippocampus, we very clearly see content-specific activation. In those recurrent circuits, we tend to see activity associated with novelty and the formation of new associations regardless of content. And it's consistent with general encoding processes. These recurrent collaterals in CA3 and dentate appear to be optimized to encode novel items and associations. And in contrast, in this other loop, this is what we call the direct loop, the direct pathway for the entorhinal cortex, it appears to be more involved in episodic, but very specifically episodic, retrieval processes. And so in the human, I think that we've nicely replicated the kind of circuits we see in the rodent. The point I'd like to make is that the hippocampus is processing content indiscriminately. There are a number of other processes that do take place in the hippocampal circuit that I have no time to discuss, that many of you have heard of and read of, and many of you have probably performed studies: pattern separation, completion, partial cue reconstruction, remapping, item versus source memory, and there are other gradients in the hippocampus that are important. But I think that throughout this, we can say that within the hippocampus, we have the same functional circuitry regardless of the task content that comes in. So the difference between animal and human hippocampus, I would suggest, therefore, is not in how the hippocampus works, but it's in the unique information that enters the hippocampus. And if information entering the hippocampus is strongly related to human consciousness, then that's what the hippocampus is going to be doing. But it's actually doing the same thing as if it were processing a place cell. So I'm going to briefly touch on the second question that I brought up in the beginning. Why is the hippocampus, therefore, so vulnerable? And this is just a small number of the many things that target the hippocampus very specifically, exclusively, and sometimes devastatingly. Viral encephalitis like herpes, fever, anoxia, epilepsy, which is very predominant in medial temporal lobe. But I'm only going to go into any details at all in stress and aging, just in the interest of time. So there's a tremendous hippocampal vulnerability to stress. As you know, stress impacts hippocampal structure and function. But there are pluses. Learned fear responses are clearly essential for survival. They are in the rat and they are in humans as well. There are serotonin receptors in the hippocampus that respond strongly to stress, and they do so throughout the lifespan, from prenatal exposure all the way through late in life, even in Alzheimer's disease. But on the plus side, those same receptors are associated with superior memory, and we've studied this using a PET ligand called 18-MPPF PET, which is a serotonin 1A receptor a binding agent that's very, very strongly expressed, almost exclusively expressed in the hippocampus and insula. And in some preliminary work by our colleague Vladimir Keppe at UCLA, we've correlated memory performance in normal volunteers with MPPF binding, and the correlation is strikingly high. It's over 0.9. So there's a huge relationship between the density of these serotonin receptors in the hippocampus and memory performance. So it does suggest that there's a plus for all of those serotonin receptors, even though they're exquisitely responsive to stress. The other biggie for the hippocampus is aging and vulnerability to aging, and particularly AD risk. And I'm going to show this very quick study from Alison Berggren, who was a pre-doc and a postdoc in my lab and is now a faculty associate professor at UCLA and is up here somewhere. And she's examined cognitively normal but older carriers of the APOE4 allele. In the flattening procedure, we are able to also capture cortical thickness information, so we can look at structure as well as function in the hippocampal subregions. And what she found is in healthy normals, so cognitively normal, no difference in memory, older folks in their 60s, that there's thinning of the entorhinal cortex and the subiculum, which is one of the areas in the hippocampus that projects immediately to the entorhinal cortex, probably 20 years before the onset of Alzheimer's disease. As many of you know, and I think Bill Seeley already discussed in his fantastic talk on Monday, network structure is altered in older people and in various kinds of aging diseases. And my former graduate student, Jesse Brown, who's now in Bill Seeley's lab, did this network analysis study of APOE4 carriers using DTI and looked at the default mode network, which of course has gained a lot of interest in Alzheimer's disease and shown abnormal or actually decreased clustering coefficient in aging carriers of the APOE4 allele. Now, of course, in both of these cases, you could say that this is preclinical disease, and this is simply a disease process. But I have another young graduate student who's just graduating soon, Tessa Harrison, who studied unique connectivity, network connectivity in very young APOE4 carriers. I'm going to show 4 pictures of her, and you'll see why in a second. You can see she's just been a great mentee and has followed nicely in my footsteps. I swear to God that this was an accident. We never planned this once. She has been working with Ahmad Hariri in the Duke Genetics Study, and they were able to get 570 resting state fMRI subjects who were aged 18 to 22, so we could not possibly argue that these are preclinical patients. So she did a couple of different kinds of analyses, but first a seed-based analysis of the default mode and the sensory motor networks. And you can see that there are some differences in network connectivity, and almost all the differences are that there's less connectivity in APOE4 carriers compared to non-carriers from the default mode network. There's one node that is stronger in carriers versus non-carriers using this seed-based approach. But she did another analysis that looked at internetwork connectivity on a global scale. And what she found was that APOE4 carriers who are young have relatively greater segregation between networks. And you might think of that as a good thing. You might just think of that as a different thing. It's just a different way of processing information. But there is some growing evidence that there might be some pluses early in life to having an APOE4 gene. This is one study that was recently published in Neuroimage showing APOE4 polymorphism in young adults associated with improved attention and also some distinct neural signatures. And there was also a recent animal study looking at a mouse mutant model of the equivalent of an APOE4 genotype. And what they found was that these knockouts that looked like an APOE4, what would be an APOE4 human, had improved synaptic plasticity and better fear conditioning, but only in the very young mice, not in the old mice. This does suggest that there could be some pluses. So are there benefits of having a vulnerable hippocampus? Hippocampus is vulnerable to anoxia, but this region also has very high oxygen utilization, higher baseline firing rate, which allows it to basically be encoding information all the time, night and day, during sleep, during the default mode. And there's evidence that pre-exposure to mild hypoxia is actually protective of later hypoxia. In epilepsy, the hippocampus does in fact remember all new firing patterns, and the kindling model of epilepsy has in fact been a primary method of studying long-term potentiation in the brain. It's a model of learning where you put electrical stimulation into a circuit, it forms a circuit, and that circuit stays, and it remains a repetitive circuit. It's a learning. It is learning, even if it's an abnormal learning. Sensitivity to stress. Well, it facilitates memory for emotional salient events that can increase survivability. And I also mentioned that aging and genetic risk for memory loss may confer unique, though perhaps not fully understood, advantages in information processing. And there's some evidence that it may enhance LTP. This is a model of antagonistic pleiotropy that some of you may have heard about. So I would conclude by suggesting first that using high-resolution fMRI with very similar tasks and very similar methods, we can see heterogeneity in hippocampal subregions just as in the rodent. And across the content areas, we see the same processes. Here I've focused exclusively on encoding and retrieval, but we find that they have the exact same subregional anatomy. The hippocampus anatomy is process-determined, therefore, I think, but content promiscuous. It is highly susceptible. It's vulnerable to any content that comes its way. I could consider it the wimpy kid of brain structures. It will just take in whatever is there and respond to it and change it fundamentally. And this permits tremendous plasticity, and it permits tremendous response flexibility. So what is unique, I think, about hippocampal function is not at all how it functions, per se. It's unique only in the content that it receives. And just like the lower animals, it will respond to whatever content that comes its way in the same way. And if we saw Martians, we would find Martian responsive neurons in our brain as well. But I would also suggest that this unique sensitivity is the same feature. This same feature of input flexibility and sensitivity to content creates an inevitable vulnerability in the hippocampus, and that's the downside of it. So, with that, I would like to thank you all for your attention. Thank you, program, for inviting me here.